Hi, this is Madeline. Cold call. Today is Tuesday, November 19th. I am doing my video finally for this week for bacterial morphology and staining techniques. Um, it's supposed to be the week that I got everything in line, but of course something came up, so I apologize that my lab is late. I already got the other lab in process, so it shouldn't be too much of a problem getting everything on time. Basically, what happened was is someone at my clinic didn't receive their certification, so they're no longer with the clinic, and someone had to come in and do the opening procedures and get everything prepped. And unfortunately for me, that was me, which means I was running a 2:30 to 6:30 shift a few days this week. And on 12-hour days, I'm usually good. I have a few hours of work left in me. I can get stuff done afterwards, even if it's only a little. But when we start creeping into the 16-hour days, it sucks. So because I had to learn how to do the opening procedures, which they should have done with me before. But anyways, stupid long hours are done. No more 50 plus hour weeks. I I already had notified them that I was taking another class, so it shouldn't happen again. Anyways, this lab was really interesting to me because I like the idea of, or I guess the procedure of doing the gram stain in order to identify, whether bacteria is going to be gram negative or gram positive. And it was very interesting to see how the different dyes worked with the gram positive and gram negative bacteria. So the it was cool to see the gram negative bacteria not being stained while the gram positive bacteria was actually stained. And what that means for the healthcare workers or lab technicians who are reviewing this uh, is that we can quickly identify, what kind of antibiotic we need to be treating someone with. Obviously, there's some more work that goes into it besides just, hey, this is gram negative, this is gram positive. There's, some identification involved before we just start sending out antibiotics willy nilly. But how quickly the procedure could be done and then interpreted was very interesting. I work with people every day, and one of the things that they have potential problems with is bloodstream infections. And I don't have the knowledge or actually the training. I draw blood cultures, when instructed and when there's a potential that they have a bloodstream infection or something like that. But I don't do the interpretation of any results, or I don't know the procedure of how they review the blood cultures. But I thought it was interesting, and it made me think about my line of work, since I do a lot of blood draws every so often. I do mostly any of the blood draws I mostly do is for labs to check our patients' values. I'm in dialysis. I'm a dialysis technician, so that's part of me moving into healthcare was finding a job in healthcare. So that's what I do. One of the difficulties that I had in this lab was the cheek cells. They stain with the direct simple stain. They stain so fiercely and intensely that it was difficult for me to define, any real specific shape that I could feel confident saying this bacteria is coccus or, whatever shape it might be. So that was kind of frustrating. It could have been in the way that I prepared the slide. Another of the difficulties that I had was which one is it? One of mine, the Staphylococcus epidermidis, that one appeared to stain both purple and red, which would mean it's gram positive and gram negative. I wrote this in my reflection, but I think that unless the bacteria stains that way typically, that it might have been a contamination either from the broth or when I did prepare the slide. Pretty careful. Well, not pretty careful, but I'm very careful about when I'm doing everything and making sure I'm using aseptic technique, sterilizing anything that's new or that has to come in contact with the broth or the test tube, for example, putting it under the flame. So I found that very interesting, and I'm not Not quite sure what went on there, or if it was my staining technique. I made sure I waited each time. Um, but that was pretty much the extent of it. I just found this lab very interesting. I'm hoping this week that I'll be on time with everything, and I don't want you to think that I'm not serious about this class. I just need to make sure they stop scheduling me, and unfortunately, they keep scheduling me, and I'm getting it fixed. That is our lab video for this week. I will see you next week.